Thanks, it's great to be here and to speak with you. I thought what I do, and I'm delighted to be interrupted at any point, I should say. I thought what I do is tell you something about some of the work that we do and what it means to be a scientist, something which has taken me many decades to figure out, although I've been one for many decades. I called this At the Edge, a scientist's story of research and funding, because I feel like I am at the edge. I am full of stories. I could sit and tell you stories about what I do all night, but I'm going to be very pointed and tell you a story about what it is to be a scientist and what it means to work in science. And I think the thing that is hard for some people to understand is that there are really zillions of great jobs that use training in science. And what I do, doing research, is just a tiny fraction of the jobs that are in science. Anyone who's working in science has already put themselves in the bottom 50% of the unemployed in our country. There are many, many wonderful jobs in science. Research scientist is something I kind of defaulted into, and this is what I do. This is what it feels like to do research. It's an extraordinary feeling of standing somewhere on the edge of a cliff, and you're not exactly sure where to go. After a while, you think that there's probably a path down, or that if you take a flying leap, there's probably a parachute on you, or there's a boat at the bottom, assuming that there's water under the clouds, or somehow, the cliff will extend itself and you can just walk along. But sitting in my office at work, speaking to people, reading the literature, thinking about what's important, what's interesting in scientific research at the frontier, where one doesn't know very much, feels like standing on the edge of a cliff and leaping off. The point of what I do is to ask interesting things, to ask questions that haven't been asked before, and to figure out whether the answers are worth having, whether they're worth knowing, whether they're important. And I thought I'd tell you about one question that we started asking a long time ago that no one else was asking, and it seemed interesting, and it turns out to be important, although we're still standing on the edge of the cliff and walking out and out and out, and so far the cliff has extended itself very kindly as we have walked. And the question is, why is our brain full of fluid? Your brain and mine has about 1.5 cups of fluid inside it. That yellow stuff contained within these things called brain ventricles. And this fluid moves around in your brain. It's replaced about 5 times a day. And it's called cerebrospinal fluid. And we started asking, what does it do? Why is it there? So I'll tell you a bit about why there's fluid in our brains. It turns out from looking at medicine and looking at human patients that if you have too much fluid in your brain or too little, it's bad. So these are MRIs of your brain, okay? And the black stuff here is the fluid. This is a patient, and this is someone who doesn't have a particular disorder. This patient has a huge excess of fluid, cerebrospinal fluid, and this condition is called hydrocephalus. You see it in babies, you see it in adults. If you see it in infants, it's very often correlated with intellectual disability and too few nerve cells in the brain. If you see it in adults, it often presents as Alzheimer's. And in fact, about 15% of patients who present with Alzheimer-type cognitive dysfunction, gait dysfunction, things that really would classically be put in the Alzheimer's bucket, have got too much fluid in their brain. If there's too little fluid in the brain, this can be devastating. As a fetus, a human fetus whose fluid leaks out will develop without any of the forebrain. That's called anencephaly, and it's incompatible with life. If there's a hole in the tube that forms the brain, further back down, the brain and the spinal cord being connected, if the tube doesn't close up in the spinal cord and fluid leaks out, you get something called spina bifida. You can't walk properly, and there are other issues. So we knew from clinical practice that there's something important about this fluid. And what we understood, because my lab thinks about embryos and the beginning of life and not about adults, what we understood is that the brain forms from a sheet of cells that rolls up into a tube and then changes shape and gets bigger and looks like a brain. And the tube of the brain forms very early when you're an embryo, and it stays there, and it's filled with this cerebrospinal fluid. And what I'll tell you, because we've just published the work and there's kind of a nice story behind it, is that my lab has just shown that the cerebrospinal fluid is needed for the brain cells to survive. And it's needed through a molecule called retinoic acid, which you may have heard of because people put it on their skin, makes your skin look good. It actually has a very strong chemical reaction on your skin and makes your skin cells grow in certain ways that remove wrinkles and things. And it's also can be a very good anti-cancer treatment. Retinoic acid is absolutely essential for life, but for the first time we've shown that it's essential for survival of brain cells working through the cerebrospinal fluid. Okay, so now let me tell you about the system we use. We use something called a model. A model means we'd like to study humans, but we can't for reasons of ethics and because human embryos develop, of course, deep inside the mother. So we use a proxy. The embryos we use are called zebrafish, and I'm going to just start some movies so that you will be able to see how these embryos develop. And one of the things about them which is really awesome is that they develop very quickly. The stuff moving on top are cells dividing, and these cells eventually will spread out to cover the whole embryo. Some of them will move to one side. They'll start to form eyes and a brain. They'll start to form muscles, your fish fillet. And within about 19 hours after fertilization, there will be an animal that's got a brain. It's starting to make its cerebrospinal fluid, and it's where a human embryo would be in about 6 weeks to 2 months. So this is very, very quick. And these are fantastic embryos and animals with which to do our studies. This is the best part. This is the story. This is the story of the people who've worked with me on this project for now a decade. It's a story of how we stood on the edge of the cliff and we said yes, and we kept going, and we worked really, really, really hard in a kind of positive loop, keeping going and keeping going. When I came to Whitehead Institute in MIT, I'd never actually seen a zebrafish embryo, but I had heard that this was a really good system. I knew frog embryos really well. I've worked on frogs since I was an undergraduate, and they're fantastic. But I'd heard that zebrafish were a great system, so I thought I would start working on them. And I told people I was going to work on zebrafish, which is, fine, but I'd actually never seen them. So these two people at the top there, Charles Sagerstrom and Zhenya Grindblatt, said, okay, we've never seen zebrafish either, but let's get some and start working with them. And so we did. They were postdocs with me, and we started working on zebrafish, and we had some specific assays in mind, and we all learned the system together, and it was very terrific. And after a while, this person joined my research group. Her name is Laura Ann Lowry, and she was really fantastic. She was a graduate student, visionary, hardworking, and we spent lots of hours thinking about what a key problem would be for her to address for her thesis. And I have to say, when I put this slide together tonight, I got very emotional putting all these people up here because we have spent so much time together. And I emailed Laura Ann and I said, Laura Ann, I got very emotional putting this slide together because I was thinking back on how we started this project. Laura Ann is now an assistant professor at Boston College and doing really fantastically. But Laura Ann and I together started asking, why is there fluid in the brain? Why does the embryo have a tube filled with the cerebrospinal fluid? And no one else was asking this question before. We just went ahead and asked it, especially she just went ahead and asked it. And from her work has come a whole project. The next person to take it over was called Jessica Chang, and it's her work I'll tell you about tonight. And then at the bottom are Ryan and Christian, two postdocs who are working in my lab now on this project. I've had hundreds of people in my lab over the years. There are additional people who've contributed on the left. There are many undergraduates, supporting staff. There's the whole milieu of the lab where there's this kind of hardworking, visionary, courageous atmosphere where we spend lots of time together and get to know each other very well and try to make something about answering questions at the frontiers of science. So these are the people, and let's now see what we learned. Okay. With all the vision and the courage and everything, you know, we wanted to get somewhere. And the place we wanted to get was to find these things called brain tube mutants. These are pictures of zebrafish embryos. And the cerebrospinal fluid has been injected with a red dye. Laura Ann figured out how to do this. It was a tour de force technique. On the top left is the one labeled WT, that's a normal brain, and all these other ones are messed up, as you can see by the messed up red, okay? There are single genes whose function is not normal in each of these embryos, and they've given these abnormal brain fluid patterns. And from them, we thought that there were clues about what the brain fluid was doing. So Laura Ann went ahead and she found these brain tube mutants, and on the way, we did some art. Those images that Laura Ann got by injecting the brain of the fish with red dye were so iconic, they were picked up by the field, the zebrafish field, immediately as the way to look at the cerebrospinal fluid. They were so iconic that some years later, I contracted with Iris Sonnenschein, who is an extraordinary quilter, and she made me this quilt, which is on my office wall, which represents Laura Ann's brain ventricle images. There's the normal ones, and then there are the mutant ones. And Iris put these together as a kind of quilt that looks like a fish tank, which we thought was fantastic, and a fantastic joining of art and science. So the goal is to understand what the cerebrospinal fluid does and to use state-of-the-art techniques to do so. This is a movie using a state-of-the-art technique with a microscope called the light sheet microscope. The light sheet microscope can take a little fish in a drop of agar, kind of jello, and it suspends it with 6 lenses around it. And it can make movies of this fish actually for days. And what you get out are movies that are huge. They are terabytes. Okay, they do not fit on any disk or drive. You have to have a special storage for them. But from this, Ryan Fame, a postdoc in the lab now, has been able to reconstruct the zebrafish brain ventricular system, the tube that contains all of the cerebrospinal fluid. It's in green here, superimposed on red, which is the tissue of the brain. And she can do this in 3D and 4D and get unprecedented detail about what the whole system of tubes looks like. She can also image how the fluid is moving in the embryonic zebrafish brain. She uses a technique where she fills up the brain with a green dye, and then she gives a little pulse of ultraviolet light. And where the light hits the green dye, it converts it to a red dye. And then you can watch the little dot of red moving from one place to another, and you can look how fast it's moving and whether it's moving uniformly from top to bottom of the brain tube, and get a sense of how the fluid might be moving through the brain. Okay, so those are some of the state-of-the-art techniques. But I go back for a few minutes to the work that Jessica Chang did when she was a graduate student coming after the beginning of the project, where we started asking this question of what the cerebrospinal fluid actually did. And what Jessica did was to develop a technique where she could put a tiny glass needle into the embryo, which is about 1/5 of a millimeter long, and pull out the fluid. So if you look at this, there's a movie playing. There's a kind of a diamond shape. That's the ventricle, the part of the tube right up at the front of the forebrain. And she's got a cannula, a little glass tube in there. She's pulling it out, and you may be able to see the diamond collapsing. Jessica went, and over many hours, she pulled out the fluid over and over again, and then asked what happened to the fish over a period of about a day, and what happened to its brain. And what she saw was something extraordinary. She saw a couple of things. Actually, firstly, the fish did not look so good. You know, the fish that the fluid had been pulled out of looked littler. They were kind of a bit bent. And more important, I think, if we have the next one, the cells in the brain started dying. So this is really right after she finished pulling out the fluid. You can see on the left it says unpunctured, that's a normal brain, and on the right it says drained. And the green cells are cells that are dying. Okay? They would normally become nerve cells, but now they're not going to become anything. And if you leave these fish for a longer period of time, you see more and more of these dying cells. You see a massive death of the brain cells. So this was fascinating to us. Jessica could figure out it wasn't that she had just pulled out the fluid. It wasn't like it was just any old fluid that would make the brain okay. It actually had to be the right cerebrospinal fluid. Okay? And in fact, she could put mouse cerebrospinal fluid back into the fish brain and make the fish brain better. So we knew there was something in mouse fluid and fish fluid that was keeping the brain cells alive. And Jessica decided to see what that was. So here's what she did. She took 500 zebrafish embryos, they're little, okay, 1/5 of a millimeter. And the brain is about, you know, 1/5 of that. And she put in her needle, and she sucked out a nanoliter, tiny, tiny drop of fluid, and she was able to put it in a tube. And then she did the same thing for the next fish, and she did that for 500 fish embryos, and then she did it another 2 times. And she eventually landed up with 3 sort of small drops of cerebrospinal fluid. She separated, and she looked to see what was in that cerebrospinal fluid. And what's in there is a lot of different proteins, okay, one of the major biochemical building blocks. And she could separate these proteins by size on something called an acrylamide or a page gel. And if you look at this is what it looks like. You put on a stain so you can see the sizes of the proteins, and this thing called a ladder kind of tells you where things run. And she put some lines across that would separate the proteins on the gel according to various size classes. And she took those pieces of gel, it's kind of like Jell-O, a thin kind of slab of Jell-O, and she could wash the proteins that were in this Jell-O out into another little tubes, and then she put them through a technique called mass spectrometry, which can actually, it's like magic, it can identify all the different proteins in each of these size classes, okay? And what she found from this, in this whole mix of proteins, in these little tiny drops from the fish, she found 378 different proteins, okay? And you know, she said to me, well, I think the best thing to do with these proteins is to figure out which of them is necessary for the brain cells to survive. I said, okay, Jessica, you know, I guess you got the rest of your life. Right, a long thesis. So this is an organization of some of these proteins. It's a snapshot, okay? And here they are. Here's some of the names of these things. And the one that turns out to be important is the one with an arrow. It's called retinol binding protein 4. So, as I said, Jessica, pulled out these 378 different proteins from the cerebrospinal fluid. And what was interesting, although she did this from fish, you can compare these proteins from human, where the same experiment's been done, and mouse and chicken and so on, and 175 of these proteins are found in our cerebrospinal fluid, in mouse, chicken, rat, etc. So those ones seemed like they would be most important because they're found everywhere. And retinol binding protein was one of them. So Jessica started down her path of asking which of the 175 proteins, she made it simpler, was the one which was really important for survival of the brain cells, and when removed, made the brain cells die. And lucky for her, because she was smart and strategic, the third one she tried was the right one. Okay, so she graduated and, all was good. Okay, and the third one she tried was retinol binding protein. I'll show you what it can do, or what something related to it can do. In fact, what she could show was that she could take the fluid out of the brain and she could put in this thing called retinoic acid, which is the product of retinol binding protein action, and she could then get rid of those green cells, those dying cells, and the brain looked much better. So this is a brain with no cerebrospinal fluid, but some retinoic acid injected back into it. It's the kind of experiment you can only do with a zebrafish, and it's a really fantastic experiment. So she could make the fish better with retinoic acid, and I won't show you all of this. She could also inject the retinol binding protein 4, and she could make the fish better with that too. Turns out retinol binding protein 4 is made by the mother, it's made by ourselves, and when someone is pregnant, it's transported across the placenta and it gets into the embryo and it takes itself up into the brain. In the fish, it's made in the yolk cell, which is the equivalent of the placenta, and it's transported from the yolk by some magic process up into the brain. And we could make the fish better after we removed their cerebrospinal fluid by injecting the retinol binding protein into the yolk of the fish, and it would make the brain better. Okay, so that was pretty fantastic. So here we have, this is called a pathway. It's a sequence of things that occurs when biological signaling processes happen. This is called the retinoic acid survival pathway, and it doesn't matter what all these components are, but retinol binding protein 4 is kind of the beginning of a whole sequence of things that leads to an outcome that somehow keeps the brain cells alive. And we don't understand the process quite yet, but we do understand that it takes place. So I wanted to show you this because we just published it, and it's taken a long time to get these data out there, and they're really important. I've shown you that in the embryo, you need this retinol binding protein and retinoic acid to keep the brain cells alive. How about in the adult? How about in an Alzheimer's patient? What if you injected retinoic acid? Would it keep the brain cells alive? We don't know. What about in other neurodegenerative disorders? Would it keep those brain cells alive? We don't know. But it's a good candidate for us and others to start trying and playing around with and seeing if it has some clinical efficacy. It's only the beginning, We started with this funny question, why is the brain full of fluid? But it's only the beginning. This is how I see research. It's a city block that's under construction. And it's really interesting. There's something really interesting going up. You can see that already, but it's not actually clear what it is. And it looks like it's going to take a long time before it's finished, which it is. But that's the idea. I've pretty much told you what I wanted to, and we could have a conversation about what you want to know. But I did spend a lot of time over the last few weeks writing a grant proposal to continue this work, to continue other work. I just submitted one today. It was noon, and we were kind of due with the grant, and I discovered we were 1 page over the length, kind of spaced out there. So that was exciting. We got 1 page off in an hour. It's amazing what you can do when you squish things and take out the line spaces and make things smaller. You know, it was fun. You know, it was exciting. Anyway, I do a lot of writing for research grants. We all do. And I would say it is a constant struggle. It's constant work to find research funding. It's one of the things about doing research at the frontiers. You find the question, you find the people to work with you, you find the money to do the work, you have the vision, you have the courage, you leap off the edge, and it all has to somehow come together. There's a real research funding crisis in our country that we who are in frontier research are all very aware of, and if you like, we can go there. But I'm going to end and stop my presentation and then have a discussion with you about what you might want to know. So thank you very much.
You were looking for a single protein that could work. Why were you looking for only one protein? Because after 3 trials, she found something. What if she continued looking for, looking at other proteins that could also work? Would that be important information for you, or?
It's a great question. You know, how do you actually deconvolute something that's so complicated? And really the only way you can do it is by trying to find some kind of simple hypothesis to work with. Otherwise, you know, it's overwhelming. So if we could have thought, maybe you need some combination of 50 different proteins. Yeah? That is, one could think about that, but it's a horrendous proposition to try to find that combination. And so, yeah, would she have found others? Yeah.
Maybe she would find another one that would be useful for other applications.
Yeah. Yes, I see your question. Yes, so that's also an excellent question of, you know, if she had continued looking, would she have found another protein which could have done the same thing? Yes. The answer is probably yes, and in fact, Ryan Fame and Christian Cortez Campos are taking that exact approach right now. What they've done, though, is to extend the assay. You know, you ask an extension of the question with a slightly different assay. What they have done is to drain the fluid for a long, long time, much longer than Jessica did. And in that case, we see a part of the brain called the optic tectum, which is one of the relay stations for vision. We start to see it degenerate completely. It undergoes massive cell death. So they are testing a whole cohort of proteins for survival of this particular brain region. So we're extending the question, but we're asking it a bit differently. And they have got a scheme whereby they've actually picked out, won't go into it right now, but they've picked out a cohort of 25 different proteins, which they're testing separately and perhaps together to see if those work. It's, you know, the process is iterative. You ask one thing and then you ask it again slightly differently and again, again, and again. Yeah. Yeah.
Jessica did really beautiful work. Jessica did really beautiful work. I don't know her personally, but I just probably expected as you did. So what I'm curious about is all these interesting neurological phenomena that people want to explore, whether it's traumatic brain injury or Parkinson's or Alzheimer's, the many animal models for this. So how involved is it to do this? And have you figured out shortcuts to collect CSF to actually analyze disease states using the zebrafish model, zebrafish embryos?
Yep. Yeah, so can we use the zebrafish as a model for specific human brain disorders? And the answer is, we think so. I would say half my group works on human disorders using the fish as a model. In fact, the approach that I alluded to has been to look at particular proteins that have been implicated in human disorders, okay, that are found in the CSF, but it's really not clear how they've been implicated, okay? So we know in Alzheimer's, for example, a protein called ApoE4 is the marker, it's the diagnostic for Alzheimer's. It's found in the CSF, okay? It's not clear what it does there at all. So that's one model we're working with, okay? It's extraordinary, actually, what you can get a zebrafish to do. We just finished a review where the last little paragraph said, the zebrafish is not a little human, but then neither is a mouse. But it's extraordinary what a zebrafish can do. The grant I submitted today has to do with a whole range of neurological disorders and body size disorders that map to a particular group of genes in the human genome. Some of the things that are wrong with the fish are seizures, anxiety, curiosity or intellectual disability, obesity. All of these things are the markers of the human disorder. We can copy each of those symptoms in the fish. It's extraordinary. So we have fish which are very anxious. We have fish with a huge propensity for seizures. We have obese fish. We have fish that appear to be intellectually, not all there. And it is extraordinary to be able to look at the fish and to use them to mimic or to mimic aspects of the human symptoms. And so that opens up being able to look at some of the devastating human disorders using the fish and all of its various attributes. Yeah.
Well, it seems like you're partly saying that there's a struggle over the scientific method, broadly speaking, in our society, and that's part of the question around funding. Do you think you could talk about that a little more?
Your question is there a struggle over the scientific method?
Well, is that part of the reason why we're having trouble getting the funding? People not really understanding that.
Well, I'll tell you my historic perspective on this is that it's always been a struggle to get research funding to ask questions of exploration. Being a scientist is being an explorer. My edge of the cliff model was really real. You have to go and explore. And I think any explorers have always found difficulty in getting funding. It's why, explorers of the past teamed up with wealthy families so that they could get their ships across the ocean and go around the world and, discover new countries and so on. And I think the analogy is really similar for scientific research funding. It can be hard to understand why one would want to go and explore without knowing what you're going to find. And that's always a kind of a question that people in charge of funding struggle with, because the people who are in charge of funding have a short-term vision, and the outcomes of exploration are often long-term. I could have given you a different talk that I just gave to a group of prospective donors to scientific research in New York, where I talked about the power of basic research. And the bottom line is that to understand how to find cures for cancer, one can lay out very clearly the kinds of foundations that have come from this exploratory research. And it's not done, actually I calculated, it has taken millions and millions and millions of research hours to understand the kind of foundation upon which we can even think about cures for cancer, okay? But it's hard to think about that prospectively. You have to have someone who has vision and so on to do so. So I think that that's really the struggle, and one needs leaders who really are willing to understand that exploration always yields. It's a win-win, but it may take longer than 2 or 3 years,
There's no other questions. Thank you so much, Dr. Sive.
You're welcome. You're welcome.
The reason to plant trees in the city is because of mitigating the urban heat island effect and reducing what the energy consumption in cities would be. If you want to keep canopy cover in cities, what I think, and thought about this for a little while. What I think we really need to do is we need to keep planting, but more importantly, we need to keep those big trees alive.